Okay, so this is a question and answer / review for chapter 5 in particular, but I would certainly take questions that you guys have all the way back to chapter 1. What I wanted to just really briefly show you is that I mentioned before I hit the record button, the notes for chapter 5 are loaded in the module, and they are, I think they're available as a PDF so that when you download them, you should be able to download them on more devices, and you can edit them, you can type in them if you want. So this literally follows the outline of chapter 5. Types of chemical reactions, and then a section on carbohydrates, a section on lipids, section on proteins, section on nucleic acids, and that's the chapter. What I would tell you is what you need to focus on for this chapter are just obviously you have to know these types of reactions, but overall there are 4 large groups of organic molecules: carbohydrates, lipids, proteins, and nucleic acids. And for each of those molecules, you need to be able to visually recognize the building block, or what we call the monomer, and the polymer. And the building blocks, the monomers, and the polymers usually have different functions. So you're going to need to be able to recognize them and then tell me what their functions are in cells. The whole chapter is just examples of this, the whole thing. So let's start with carbohydrates. Just real briefly, I'm just going to walk you through the carbohydrate one so that you can do this on your own. And I would encourage you to watch the videos from the summer, because that's exactly what it is. It's me with the pictures from the textbook going through this particular lecture in 2 parts. So for carbohydrates, the visual clue for you is we abbreviate carbohydrates as CHO, all capitals, because they mostly have carbon, hydrogen, and oxygen. And it's usually twice as many hydrogens, and the carbon and oxygen is usually the same amount. So a lot of times you'll see people abbreviate this CH subscript 2O, because it's usually twice as many hydrogens as carbons and oxygen. And the monomers for carbohydrates, I think the rule is to be a carbohydrate, you have to be at least a chain of 3 carbons, okay? So that's not very many. If it's a chain of 3 carbons, it's usually drawn as a line or in a linear form. Once you get to 5 or 6 carbons, which are the more common carbohydrates, monosaccharides, the monomers of monosaccharides, you'll see them in a ring form. And I did put on my chalkboard behind me, and I'll move things over so we can see them in a few minutes. I did put a ring form of a 5-carbon sugar, and then a linear form, and then the ring form. This isn't chemistry, so you do not have to be able to convert the linear form to the ring form. Don't care. You need to, and I would say with about 100% certainty, you need to know the ring forms. You need to be able to recognize the ring forms. The challenge is, though, these ring forms, I'm going to jump out here and go back to see if I can find those PowerPoints. The ring forms have they abbreviate the structure, and they stop writing the carbons in the ring structure as it goes along. So try to minimize this, and I guess I'll just have to go to those actual pictures, which would be where, if they're not in spring. Yeah, I don't know where I put those. I guess they're just on the website. I was trying to avoid logging into Canvas first thing in the morning. So actually, we'll just look at the picture on the board here then. I'm going to do this this or not, but we're about to find out. I think, and then especially if I had some good, especially if I stand back here, I think it tries to focus on what's back. Right? So this picture here is the linear form of ribose of a 5-carbon sugar, and see how all the carbons are written out. When they switch to the ring form, this is the exact same monosaccharide. This is the non-abbreviated ring form, and I get it. There's glare. There's a little bit of glare on that. I get it, but it's the best I can do. This ring form, notice you can see the oxygen, the carbon, and all the hydrogens written out. The abbreviated ring form, they're going to drop the carbons, and they're just going to draw it like this. They're just going to put an O, and they're just going to leave lines like that. I know it's a crappy drawing, sorry. This is the abbreviated form, and the abbreviated form, you have to remember that every place where 4 lines come together, 4 chemical bonds come together, that's understood to be a carbon. That's the tricky part about the abbreviated forms. If you're following along in the book and you're just like, what happened to all the carbons? She said, count the carbons. I don't see any of the letter C to represent carbon. That's what happens to them. This is carbon 1, 2, 3, 4, and this is the 5th carbon. We count them clockwise after the oxygen. Okay, so the oxygen is kind of like the 12 o'clock on the clock, and then post the oxygen clockwise, we count this as carbon number 1, 2, 3, 4, and the 5th carbon is out here. Whereas in the chain, it's really easy to see because they're just in a line. You can just count them, right? So that, I think, is the tricky part about carbohydrates, about monosaccharides, and the structure portion. The function of monosaccharides primarily just be an energy source for cells. And of course, when you string them together, you can make a polysaccharide, and then the polysaccharide is going to have a different function, right? So the polysaccharides are, you can use them to store energy, but you can also use them as structural molecules in cells, and they are used. So cellulose you might recognize from plants, cell walls. Peptidoglycan is what's in the cell wall of bacteria, and chitin is what's in the cell wall of fungus. Now, fats, I think I drew a fat on the other side of my board. So let me go back. Oh, I should make this one, it's not so much gymnastics. So a fat, or in particular, I guess we should say a lipid. Lipids come in different varieties. The main thing you look for if you're trying to figure out if something's a lipid is you look for glycerol and 3 chains of fatty acids. And sometimes those chains of fatty acids will be substituted with rings, like for example, if it's a steroid, or with a different group if it's a phospholipid. But if you look for those long chains of fatty acids, that's the big clue that it's a lipid. Okay. Lipids primarily function as energy storage and insulation. And again, later I'll flip that board over and we can take a look at a picture of a lipid. Proteins are the most complicated of the bunch. And I think I'm just going to break down and go to the pictures because I can't draw all the things I want for proteins. So if you guys can just be patient with me for a second while I log into Canvas. looks like we've purchased more bandwidth. Yeah, access denied. Okay. The reason I can't see my, okay. Yeah. All right. So today it says I don't have access to my course. That's great. Okay. Yay. All right, so I'll just keep trying. Maybe eventually I get in. Okay, I don't have access to my class. All right. It's okay. All right, so let's go back to just talking about proteins then. Sorry, I don't have any pictures. So proteins are made up of amino acids, and there are 20 different amino acids. Again, you don't have to memorize all 20 of them. Go to chemistry, learn all about it. What you do need to be able to recognize is the individual structure of an amino acid, which the structure they have in common is they have a nitrogen, a carbon, a second carbon, and that last carbon in the amino acid has a double bond to an oxygen and then a single bond to another oxygen. And I'll show you a picture on the board in a second. When you string amino acids together, we call them peptides. And when you string many of them together, we call them polypeptides. And a polypeptide does not become a functional protein until this secondary, tertiary, quaternary structure happens to it. So one of the challenges with proteins is that they have this higher level of organization that then dictates their function. But the advantage is because they can be almost an endless, like words in a book, they can be almost an endless combination of amino acids. Proteins have the most functions of any of the organic molecules. Usually I tell students the short answer is proteins do everything, just about, okay? They're not great at everything, but they can do everything in a cell, pretty much. And then the last category, the fourth category there would be the nucleic acids, and those are composed of a nucleotide, a sugar, a monosaccharide, and a phosphate. And so, again, I'm just going to give it a couple minutes, and then I'm going to try to get into Canvas again, and I can show you some of those visuals real quickly. Questions you guys have about specifically from anything from chapter 5 that you've read chapter 5 and you're just like, this doesn't make any sense to me? No, yes, you haven't read chapter 5 yet. That's okay. Anything about the homeworks, although now I can't get to them, but I'll try again. Let's see if I can see if it'll let me in now. The basically the whole college logs in between 8 and 8:30, and so sometimes that's what it is. Okay. Yeah. Okay. Maybe this time. Fingers crossed. Okay. Well, I won't be working on my course if I can't get into it today. All right, so I have a couple more organic molecules on my board, and I'll flip the board over and we can talk about them. And if you would have to recognize them, there are actually pictures on the exam. And I believe I have it sorted so that you'll get, I think there's 6 or 7 of them. You'll get at least one of each of the different kinds of organic molecules. So you do need to be able to recognize the pictures and recognize them fairly quickly. And then the multiple choice questions from chapter 5 will be mostly on the functions of them. Are there going to be questions about peptidoglycan? The only thing is that you might have to recognize that it's a polysaccharide. Come to microbiology, learn all about it, right? So I'm a microbiologist, so I can't not put that in there. I drew some other pre-coffee molecules this morning. I'm just going to hold this here for you now. There we are. It's a little better actually on the whiteboard. What do you guys? Especially if I stand back here. Okay, so it looks like this is the worst. This molecule up here, this I actually drew adenine. This is one of the 4. This is a nucleic acid. And the nucleic acid itself has 3 parts, right? We call it a nucleotide. This part, which is a purine, and then a ribose or deoxyribose if it's DNA or if it's RNA, so a monosaccharide, and then a phosphate. This is adenine, and like I said, I don't expect you to be able to tell me that's adenine, right? You don't have to be able to say that, but there are 2 types of what we call, actually I should have written nitrogenous base, because nucleic acid implies the phosphate, so let me erase that. Here. This is really what we call a nitrogenous base, and we call it that because it has nitrogen in the ring. So if you look at this carefully or look at the picture carefully that's better drawn in your textbook, you'll see there's actually nitrogens in the ring, whereas in a protein or an amino acid, the nitrogens are in the chain, they're in the backbone, but they're not in the ring. So a nitrogen-containing ring, nitrogenous base, they come in 2 forms. This form is called a purine, because it has 2 nitrogen-containing rings. The other form of nitrogenous base is called a pyrimidine. And it only has 1 ring with nitrogen in the ring. You should be able to tell the difference between a purine and a pyrimidine. You are not responsible to know that adenine is a purine and pyrimidines are thymine and cytosine. Okay? Not important. But you should be able to recognize and say, oh, that's a nitrogenous base and it has 2 rings, so it's a purine. Right? Function, and I have something to admit. Check what I can do from over here. Functions would be for DNA and RNA, so it's all about genetic information, storage, and transfer. This, anybody recognize this? What kind of organic molecule this one is? You've been studying or paying attention, or you've already had chemistry?
Triglyceride.
Sorry.
Triglyceride. Is it a triglyceride?
Wait a minute, I got to turn my volume up. Tell me again, dear.
Is it a triglyceride?
It is. That's very super specific. I probably won't even ask you that specifically. If you could tell me that this was a fat, I would be jumping up and down for joy, right? Yes, that's a lipid, fat or lipid. Glyceraldehyde here, this portion. And then 3 chains of fatty acids, right? The phospholipid, which is going to be probably the most studied lipid for us in Bio1, we abbreviate it looking like this, right? Where the hydrocarbon tails, these carbon, 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 hydrogen tails, And there's a double bond here, which is why it has a little leg kicking out. So it only has two. So the glycerol is in here, right at the base of this head. And only two chains are attached to fatty acid. The third chain is attached to choline and then a phosphate group that sticks out. This is a phospholipid. That's probably the most important lipid. Phospholipid, sorry. That's probably the most important phospholipid, important lipid that we're going to study in Bio1 because all membranes in all cells are made of phospholipids. And so that's going to be like a mantra to Bio1. All membranes in all cells. Chapter 6, chapter 7 is all about the phospholipid and how it makes the membrane. So all through this chapter, they're showing you pictures of things in a lot of ways of things to come. And lipids are probably a good example to talk about how, like the triglyceride is something that cells would use to store energy or for insulation. The phospholipid, they use it for something completely different. They use it to make a membrane. And then a steroid used for something completely different that's also in the membranes, but it's also can be used in some animals and plants as a chemical signal, right? For internal regulation to tell cells to do things. So it kind of depends on the different kind of polymer, and they usually have different functions. Questions, other questions you guys have? Should put another chair over here so I can sit down. I can just be small. Can we go over denaturization, please? Angelo asks. Yes, in terms of that has to do with proteins, right? So, sure. I think I'm going to erase my pictures. Now, you can't ask me any more questions about this because they're going to go away. So, denaturing or denaturization, I think it's called. Usually we use it as a verb. Denaturing. This happens to proteins. And the reason it happens to proteins in particular is because what it means is that secondary and tertiary and even quaternary structure that proteins have, because they have those increasing layers of complexity, that means that that structure is broken down, altered. So sometimes it's completely destroyed. Other times it has to do with if heat is applied, bonds are broken, or if an acid is applied, new bonds start to be formed. And for proteins, these secondary, tertiary, quaternary structure, that's what allows proteins to have their multiple functions, or functions, I should say. And so if you destroy this sort of higher level of structure, then the protein can no longer function. And that's what we refer to denaturing, right? What causes it primarily are going to be heat and acids, pH change. Acids are better at denaturing than bases. They can denature, but it's primarily acids. Or if you want to get super specific, later, if that's a protein that's inside a cell, like a protein you're eating, right? Other proteins that we call enzymes can actually break down or denature proteins. Other proteins, let's say. Does that help, Angela? Yes? No? Maybe so? Yep, no problem. We might see this in when our lab kits come, because we might be doing, I think we have an enzyme lab, and I think we might, I don't know, add boiling water or add lemon juice. The boiling water would obviously be heat, the lemon juice would be acid. Like I said, I don't really know what's coming in those kits, so maybe I'll try and find that out. I have the directions, but that's all I have. I don't have an actual box in front of me to know what's actually in there. So we'll see how well we can piece it together. I think the homework is up through chapter 5. So should be good. I will go through, I know there was homework that was due yesterday at midnight, right? Last night at midnight. So I'll try to go through and see anyone who hasn't completed that. I am willing, especially here in the beginning, to give you an extension if necessary. I don't want it to become a habit though, because like I said, there's 80 some of you, and so it's a chore to go through and find everyone who hasn't completed, but there are, I think, 2 people who still haven't gotten into mastering yet. So don't panic if you're having difficulty getting into mastering or getting into mastering through Canvas. We need to work on that today, tomorrow, and then you'll be kind of up to speed with everybody else. So just wanted to put that out there. And I don't know of any other general things that are going on. Other questions you guys have?
Could we go over cholesterol?
Sure. I knew you were going to make me draw that. I knew someone was going to make me draw that. Sure, I need a cheat for that because I can't draw it without looking, because I am a chemist no more, right? So actually, it looks like your book puts in, yeah, they put in cholesterol. So I'll draw it for you, right? So all of the sterols, their key feature to figuring out if it's a sterol is this 4-ring structure here that I'm trying to draw. Don't tell Professor Choi or Feudelli what a terrible organic chemist I am, because I had a lot of biochemistry in my day, but it was a long time ago now. There's an OH out here. And then we draw off of this last guy. H3. Go up, down, up, down. H3. H3. Something like that. Don't hold me to it. That's perfect. Right? So all of the steroids, so this would be an example of a steroid, that is a subcategory of lipid. And so it's a special type of lipid, like the phospholipid. It's specialized in its function and its structure. The way you recognize a sterol or a steroid is by these 4 rings. And notice there's no space between them. They're all part of the other ring. This is different, and visually what will help you keep it separate from a polysaccharide is a polysaccharide will have, let's say this is, they will, when polysaccharides are joined together, even though they're rings, when they're joined together, they're always holding hands with oxygen, I like to say, right? So there's always this space, hydrogen there, where the 2 rings, this would be a polysaccharide or the beginning of a polysaccharide, right? And they always have this either up or down, depending on the orientation of the sugar, which again isn't important for Bio1. They're going to have this gap where they have this bond with oxygen, Steroids, no. See how all the rings are just they're right part of the next ring. So we say they're 4 rings tightly bound together. And then what varies is which kind of ring and how long. This is the chain of fatty acids. It's a lot shorter than in the triglyceride. And because of this 4-ring structure, they're very stiff. So what they are literally used for, mostly cholesterol, ergosterol, a lot of them are used for 2 things. They give cell membranes, and sometimes they're in cell walls as well, but predominantly cell membranes. They give cell membranes a certain rigidity. So phospholipids, right, that we had drawn before, like phospholipids, when they make a membrane, and there's probably a picture of this in chapter 5, right? So and we talked about it a little bit last week because someone asked me about negative charged heads and the lipid tails, right? Phospholipids are squishy because this is the lipid portion. So it's just fat against fat. So it's just like, coconut stomach or whatever. Cholesterol gets inserted in these membranes. And I'm just going to draw it rather badly here, but cholesterol would get inserted like here. I'm just going to draw 4 squares. And that helps give that membrane a stronger structure or shape. So it's not quite so flexible. The second function of steroids, obviously, is like we said before, they can be hormones. Okay. And the job of a hormone is to signal other cells, right? To tell cells to do something, to move in a bloodstream or in a case of a plant through various vessels and tubules to get from one cell to another to tell those cells, the second set of cells, to do something, grow, produce a hormone, produce another chemical, whatever it is. Better, worse? Okay, great. Probably more than you wanted to know, sorry. It's exciting to me sometimes to think about actually talking to you in a classroom, right? And telling you all these things that would be interesting to know instead of just what we have to know. Which is the boring part, but unfortunately. Anyone else? Any questions about peptide? Okay, no. I would say the first test is rather balanced with the least amount of emphasis on chapter 1 because there's the least amount of information in chapter 1. But then for chapters 2, 3, 4, and 5, it's not 4. For chapter 4, obviously, it's just that chart, that table. So I think I forgot how many you get, but it's 2 or 3 off of that chart. You just have to identify them. And chapter 5, like I said, will be structure and function. There's probably the most questions from chapter 5. So that's probably the one you should be the quickest at. I'll see you on Monday though, right? So we can talk again then.
Is chapter 6 going to be on the test?
I don't think so. I think this first one, I used to do 1 through 7 when we were in person, but online I found that it's too much, it's too big of a bite for most people. So we're just going to do 1 through 5 on this first one. Is that okay?
Okay, yeah, I have one more question.
Yes.
With the anti-parallel structure with the 5 to 3, do we need to know exactly what that is? Because I can't.
No, not today. Not until chapter 16-ish, maybe.
Okay.
Did I ramble about that in the video?
No, not really.
Okay.
I was just for myself.
Okay. More than you need to know for chapter 5. If you can remember the 4 groups, what the monomers look like, what the polymers look like, and the functions of the monomers and the functions of the polymer, that's plenty. And the 4 levels of protein organization or protein structure and the list of functions for proteins, you'll be good.
Okay, thank you.
Mm-hmm.
Yep. And then once we get this boring part behind us, then we go do some, like when we learn about cells and other things that will get a little more interesting, hopefully. Anybody else? Yes, no, maybe so. All right, then I'm gonna stop recording and